Good afternoon, everybody, and welcome to this afternoon session of our discussion meeting. Today we have Professor Collins Assisi from IISER-Pune speaking on the neural encoding of odors in a turbulent environment. Collins?
Thank you. So first I should thank the speakers for the organizers for the opportunity to speak here. I'll be talking about olfactory processing, and the work that I'll be describing was done with my collaborator, an undergraduate student at IISER-Pune, Rishika Mohanta. She not only drove many aspects of the research questions, but also built the simulation tools from scratch. So we ended up using Python and TensorFlow on GPUs to integrate largish arrays of coupled differential equations very efficiently. So you can download these tools off my website or of Rishika's GitHub page. In fact, I'll try to put it up on the resources listed in the meeting website. So anyway, olfaction with other chemical senses is phylogenetically perhaps the oldest sense. And in fact, as far as chemosensation is concerned, bacteria can track chemical gradients. So the problems that are solved by the olfactory system, for example, feature detection, where you have to tell different attributes of an odor, like its concentration, background suppression, selective attention, where you have to attend to one particular odor and ignore stuff in the background. Also, how prior experience of odors modifies our perception of the odors that we are smelling. All of these problems that are solved by the olfactory system are also solved by other sensory systems, such as vision or audition, right? So understanding the mechanisms underlying olfactory perception will likely provide us insight into a successful and maybe even an optimal algorithm, a biological algorithm for processing complex information, right? And the goal of this talk is to try and think about how this algorithm is implemented in the wetware of the brain, right? So the tasks that the olfactory system, the goals that it's achieving, is despite the remarkable complexity of the sensory input that comes to the olfactory system. So when you look at odor molecules that you smell every day, like a lime or a rose, it consists of congregations of tens to hundreds of different molecules. And not only are the individual or the components of an odor complex, it also arrives in very unpredictable ways, like riding on plumes of air as you breathe it in. So not only is the structure of the odor complex, but also the temporal pattern that brings it to us is also very complex and unpredictable. So the question we're going to ask is, how does the olfactory system recognize odors in an invariant manner, despite these complexities and inevitable noise that you see in the environment around us? Okay? So before I get into a description of the olfactory system and so on, what I'm going to do is I'm going to put this acknowledgement slide out here, because one tends to be in a hurry towards the end of the talk and miss acknowledging people who really matter. So I would like to acknowledge discussions with people in Suhita Nadkarni's lab and with people in my lab. And some of the ideas in this project were initially started off with Maxime Bajanov, who's currently in UCSD, and Mark Stopfer at NIH. Rishika was funded by KVPY. Part of this work is done with funding from the India Alliance and lots of support from IISER-Pune. All right, so let me start off by giving you a broad description of the organization of the olfactory system, right? So this particular image is one I took from the Nobel Prize website. The Nobel Prize in Physiology and Medicine I think it was 2004, I might be completely off, went to Linda Buck and Richard Axel for their work in studying the organization of the olfactory system. So here what you have is this rather funny looking flower, which is emanating a bunch of odor molecules. These are these red, green molecules that you see out here. And this person inhales these odor molecules that go in with so the air is laden with these odor molecules, and it goes into the nasal cavity. And you have a zoomed in where we're going to zoom into this part of the system. And what you have out here, this is the nasal epithelium, and there you see a lot of these neurons. These are the olfactory receptor neurons, and they have these processes that you see out here, and these molecules waft into the nasal cavity, and these odorant molecules can go and stick onto proteins that are present on these odorant receptor neurons. So these odorant receptors, once the odor molecules bind to these receptors, depending on whether they are in fact receptive to that particular molecule or not, once they bind to these odorant receptors, the shape of the configuration of the receptor can change. And this can in turn lead to a whole cascade of events, which may eventually lead to this neuron generating an action potential. Right? And this electrical stimulus then goes on from the receptor neurons, and it goes to the olfactory bulb. Now, what's really interesting in the organization out here is that each of these olfactory receptor neurons express only one kind of odorant receptor, right? And different neurons, different receptors. Now, you'll see that the neurons that express a particular odorant receptor type is sort of distributed all over the nose, right? So it's randomly distributed all over the place. But what's really fascinating is that regardless of where it might be geographically located, all of these neurons go and converge into this spatially circumscribed region in the olfactory bulb known as a glomerulus, right? So there is this remarkable glomerular organization that you will see across species. You'll see it in insects, you'll see it in mammals, all over the place, all right? Now, this is a rather complex system with a huge number of neurons. And so one of the things that we do is, rather than look at the mammalian olfactory system in all of its remarkable complexity, we're going to look at a far simpler olfactory system. And this is the insect olfactory system, in particular the locust olfactory system. And there are differences between the locust olfactory system and, say, the fly olfactory system and so on. But we're going to talk about the locust olfactory system. There are differences, there are peculiarities, but there are also a lot of commonalities in the organization of the olfactory system across insects and mammals. All right. So here, so just to point out to some of the differences, instead of the nose, in locust you have antennae, and the receptor neurons are there in the antennae. And instead of the olfactory bulb, which you see in mammals, in the insect olfactory system, you have something known as the antennal lobe. So the receptor neurons, so the odor molecules stick to the receptor neurons, which send their input to the antennal lobe, which is the first relay of the insect olfactory system. So it's the equivalent of the olfactory bulb in mammals. And inside the insect antennal lobe, you have a bunch of neurons. So it's a highly recurrent network of neurons. There are excitatory projection neurons, and there are inhibitory local interneurons. Now, from the antennal lobe, going further down to the mushroom body in insects, and the equivalent would be the olfactory cortex in mammals. So it's only the projection neurons that project outwards from here and go to the mushroom body, right? The inhibitory interneurons have projections only within the antennal lobe. They don't project outside the antennal lobe, right? Now, this particular network, which has been, number of experiments people have looked at and recorded the activity of these neurons, it shows remarkable spatiotemporal patterns of activity as you stimulate the system with an odor, right? So what happens when you give an odor to the antenna? You waft an odor across the antenna of the insect, and you record the activity of these projection neurons. And these are recordings that were done in Gilles Laurent's lab a while ago. And, over the decades that people have recorded from the antennal lobe, I would think that of the 900 projection neurons, they probably oversampled the number of neurons that are recorded, right? So it's a very well-studied system. So, all right, so here you have the smell of cherry that is pulsed or that is sent across the antenna, and then you look at the response of 3 of these projection neurons. Now, this dark line out here is the time during which the odor is presented, and then you have a response of this. Now, there's some amount of background activity that you see, but if you look at the activity of PN1, it's inhibited. PN2 fires for a short duration. PN3, the firing rate seems to go up. If you present MINT to the same set of neurons, you'll see that the pattern of activity can be quite different for the same set of neurons. And here PN1 is firing a lot of spikes, whereas PN2 is inhibited, and so on. So there are 2 things that are important out here. One is, did the neuron fire? Was it inhibited? So what's the identity of the neuron that fired? And the second thing that's important is at what time did these spikes occur, right? So it's a pattern that evolves in time. All right. Now, if you present the same odor, right? So not only do you have the spatiotemporal pattern, but it also turns out that these patterns are reliable, as they would have to be if they were to represent an odor. So if you present MINT 2 times to the same neuron or to the antenna, and you record from the same neuron, the activity is going to be more or less the same, right? So strawberry, apple, octanol, and so on, all of it reliably generate the same set of spikes, right? So not only do you get spatiotemporal patterns, these spatiotemporal patterns are also quite reliable. Okay, so in addition to that, so one way to think about the neural representation of an odor is, you can think of it as a movie that's evolving in time, right? Different pixels are turned on, different neurons are turned on at different times. And what's the frames at which this movie evolves? Well, if you were to calculate the average activity of all of these neurons, right? Not the activity of individual neurons, but the average activity of all of these neurons, you're going to get something that looks like this. You get a rather nice oscillatory pattern, which is an oscillation that's approximately 20 hertz during the time that the odor is presented, right? So one way to think about the neural representation of an odor in response to these odorant inputs is to think of it in this way, right? So you have you have the local field potential generating these 20 hertz oscillation, and if you were to take each of these frames, meaning each of these oscillatory cycles, you're going to get a different population of neurons either turned on or inhibited, and so on, right? So the state of the entire network in each of these oscillatory cycles evolves over time, and you're going to get transiently synchronized ensembles of neurons that get activated as the odor is presented, and over time the representation of an odor is different transiently synchronized groups of neurons firing in different oscillatory cycles, right? So that in essence then is how an odorant is represented in the locust's brain, right? So that's maybe the smell of a rose. That's just one pattern or one movie that you see out here. All right. Now, so how do we sort of think about this in a realistic context, right? So if you look at the way you actually encounter odors in the environment, it's not this lovely static DC input that's going to the receptor neurons. It turns out that it is a rather turbulent, these odor molecules come in riding upon chaotically pulsed plumes of air. So if the insect were to stick its antenna into one of these plumes, it's not going to get something that's a DC input, or it's going to get something that's that comes in rather intermittently and in an unpredictable fashion, right? Now, what we have argued so far is that the time at which a particular neuron fires matters, because we've seen that when you give a DC input, that neuron fires reliably at around that time. The identity of neurons which fire, all of that is preserved, right? So it's both the identity of the neurons and the time at which they fire. However, when you encounter a pattern as turbulent as this, how would the temporal representation matter? Right? How would the pattern evolve? What would be the temporal component of the odor representation, and what are the kind of invariants that you can get out here, right? So now, one could think of this turbulent pattern as just being some noisy pattern that you have to overcome in order to represent the odor as a spatiotemporal sequence. But it turns out that this intermittent input may not be a bug; it might actually be a feature. And so to think about this, there's this really nice experiment that was done by the Laurent lab in 2015, where they have a locust and you present it with a continuous stream of input, right? So it's odor coming in a continuous stream, and it has the ability to move its antenna in and out of the stream. Now, you would think that if the insect wanted to recognize the odor, it would just stick its antenna into the stream and sample it for the longest time possible, create an odor representation, and that would be the end of the story. But it turns out that that's not what the locust does. What the locust does is it intermittently sticks its antenna into the odor stream and takes it out. So even if it's presented with a very simple pattern of input, the animal injects intermittency into its sampling behavior, such that the odor comes in a pulsed manner. And you can also think of the way when we encounter odors, if it's a novel odor, we probably sniff intermittently in order to get the odor input and make sense of that input. So it seems like this intermittency, this unpredictability of or this pulsed behavior of the odor input might not be a bug; it might actually be a feature, right? So the question that we're going to address is, what are the neural invariants of an odor? Alternatively, why does a rose smell like a rose, even though it comes upon extremely unpredictable, temporally random patterns of input, right? Okay. So in order to address this question, what we did is we built a computational model of an insect antenna lobe, of the locust antenna lobe. And the way we did this was we constructed projection neurons and inhibitory interneurons, and the properties of these projection neurons and inhibitory interneurons were such. So we introduced ion channels that would make these neurons fire in ways, in manners that were consistent with what you see in the experiment, in experimental recordings. Now, the projection neurons provide input to inhibit. So there are two forms of input that come in. One is the input that comes in from the receptor neurons. We don't explicitly simulate the receptor neurons, but we have random patterns of activity coming in from the receptor neurons. And these inputs drive both the inhibitory interneurons and the projection neurons. Now, what we know from experiments is that the input to projection neurons, so projection neurons respond to are more sensitive or are more specific to the kind of odors that they respond to, whereas the inhibitory interneurons are rather promiscuous and respond to a whole range of odors. So the input to projection neurons, so the input to projection neurons comes in from the receptor neurons. Now, the connectivity of this network is such that the projection neurons connect to the inhibitory interneurons, which in turn inhibit other projection neurons and also reciprocally inhibit each other. And as far as we know, there are few, if any, lateral excitatory connections among these projection neurons. Right? So these are the properties of the antennal lobe network that we built into our computational model. Now, the kind of spatiotemporal patterns that we encounter in the antennal lobe network are driven to a large extent by inhibitory interactions, right? So what are the kind of inhibitory interactions that we have in the antennal lobe? So the inhibitory interaction, so there's a densely connected reciprocally inhibitory network that you have in the antennal lobe. So here is the activity of two inhibitory interneurons that are reciprocally connected to each other, the simplest possible network that you can construct. Right? And what you have out here is, since the orange neuron out here is inhibiting the green neuron, when the orange neuron fires spikes, the green neuron is inhibited, it remains silent, and then at some point it flips over like a switch, and the green neuron fires and it turns off the orange neuron. Right? So inhibition sort of explains why one neuron is silent and why the other is, when the other is firing. But what accounts for the switching kind of behavior? Well, there is a calcium-dependent potassium current that is also present in these neurons, and as the number of as the neuron spikes, the time between these spikes progressively increases such that the orange neuron can no longer suppress the activity of the green neuron and it switches over, right? So that's what gives rise to this back and forth kind of competitive behavior. And if you turn off this calcium-dependent potassium current, then just one of the neurons spikes and the other is silenced, right? So these competitive interactions between the inhibitory interneurons is what drives spatiotemporal patterning in the insect antennal lobe. Now, how do we expand this sort of elemental interaction between two neurons, and how do we expand it to the whole network? Well, one way to think about it is that if you had multiple groups of neurons, that where the inhibition was across groups, but there was no within group inhibition. And if you simulate a network like this and you plot the activity of this network, where along the x-axis you have time and along the y-axis it's the identity of the neuron, and each of these little lines out here tells you that this particular neuron fired at a given time, right? So that indicates the time at which a particular neuron spiked. This is a raster plot. And what you will see is that this blue group of neurons fires together, the green fires together, and the orange fire together, and they don't fire together at the same time. So the structure of the competitive network determines which neurons fire together in a group. Now, these sort of competitive interactions giving rise to spatiotemporal patterns is not something that's unique to the olfactory system. You will see it in a lot of other systems, most notably the kind of central pattern generators that you see here, in this case, in the somatogastric ganglion of a lobster, which shows you neurons that fire at different phases. And this phase distribution is dependent upon the connectivity of the network, right? So these competitive interactions are really the engine that drives the patterning that you see in a lot of different networks in the brain. All right, now let's look at this for the specific networks that we have. So if you had 3 inhibitory interneurons that were reciprocally connected to each other, what could happen is, if you drive the system in a regime where one of the neurons were spiking, and you could give it a tiny bit of a perturbation such that the activity would switch from this red to this blue neuron. Right? And different perturbations can cause a switch in the state of the network. So you can use external perturbations to switch the state of the network, and it goes to a whole array of states depending on the nature of the perturbation. Now, we know that projection neurons drive the inhibitory interneurons. So the activity of the projection neurons could in fact serve as these perturbations that drive the network to different states. That's what we show out here, where you have projection neurons and inhibitory interneurons, and you could give excitatory drive to the projection neurons that would in turn lead the inhibitory interneurons to switch in the activity. So you'll get a rather nice pattern of sort of competitive switching between different nodes of the network. Now, these are, of course, rather simple toy models, and we know exactly what it's going to do because the structure of these competitive interactions is very clear. But what happens when you have a network like that, that's a random network, and this is the adjacency matrix of the random network, where a particular 2 nodes are connected or not, right? So that's the adjacency matrix that you see out there. And if you look at the activity of this network, where you have 30 of these inhibitory interneurons that we perturb at different points in time with short random perturbations. And you'll see that when these perturbations come in, the state of the network can flip, and it can flip in some seemingly unpredictable manner. And the question is, can we sort of bring some order to this whole thing? Can we wrap our minds around these random networks and the dynamics of these networks? And in order to do that, what we resorted to is we tried to think about the structure of these networks in terms of modules, or in terms of communities that can be detected within these networks. And let me sort of give you what I mean by this, right? So here you have a network of nodes and there are dense connections between these nodes, but there are sparse connections across. So you could think of these densely connected subgroups as modules. And it turns out that detecting modules is a rather nice exercise and a meaningful one. And here's an example that I think relates rather nicely to our to current politics. This is an example network of books, right? And these are books. Each of these nodes out here represent books, and the shape, so the circular nodes so each of these nodes are books, and the edges connecting nodes are those books that are bought by many people, right? So many people read these two books, and so you'll connect them by an edge. All right. Now, the circular nodes out here are books that have a liberal political leaning. This is an American survey, and the square nodes are books that have a conservative leaning. And you will see that this Newman clustering algorithm that detects communities in these graphs very nicely separates out the conservatives from the liberals, right? So there is some meaningful structure that emerges from these sort of community clustering algorithms. And here's another example that looks at the anatomical connectivity in the macaque monkey brain. And there again, so that's the sort of adjacency matrix that describes the connectivity of the brain. And you will see that when you sort of try to find communities out here, you'll discover that different blocks corresponding to different communities also seem to have some spatially contiguous representation in the brain. So it is a meaningful sort of a description. Now, how does this translate to our competitive networks is something that we need to think about. So in our competitive networks, notice what we have out here, right? So this group of neurons has dense, so there are dense across group connections, but few within group connections, right? And what this means is we have the opposite of a network that we have on the top out here, right? Here, the within group connections are dense and the across group connections are sparse, whereas here, the across group connections in this case are not there, but could be sparse or could be dense, but the within group connections are just completely not there. Right? So how do we discover these sort of communities that would indicate competitive interactions, right? So how do we look for these communities? Well, one simple way to do that is, let's say you're given a network, you can flip, and by flip the network, I'm saying look at the complement of the network. So all the nodes that were connected in this network, in the flipped network, are not connected. And those that were not connected, you draw an edge between them, right? So the adjacency matrix has zeros and ones flipped, except the diagonals, which are self connections. Now you find the communities in this complementary network, and you group these communities, and then you flip it back again. Right? So when you look at the adjacency matrix, you initially have a network where this is the connectivity between the inhibitory interneurons. You take the complementary network, apply the community clustering algorithm that looks for dense connections within, and you flip it around, and you will see that you get these, in this particular case, you get two groups of nodes that have sparse connections within, but have dense connections across these nodes. Right? Now, this sort of an architecture has dynamical consequences, and that's what we're going to look at in what's coming. All right. So if you look at the dynamics of this network, now here is the network where we have sort of arranged these nodes based on which community they belong to, and you'll see these large gaps out there saying that there are sparse connections within and off-diagonal blocks that have dense connections across. Now here, we have stimulated this network of LNs that's connected using this adjacency matrix. You stimulate that network, and you sort of start seeing more correlated activity corresponding to these blocks out here. Right. So what I'm plotting in this particular figure is the correlation of activity that's measured over some length of time. Right. And you'll see that it's more correlated within blocks than across blocks. So stuff that are sparsely connected tend to fire in a synchronized manner, and that sort of fits our intuition. Right. Now, if you look at the activity of this network, rather than look at the activity of individual neurons, we can club together. Of course, it's not a perfect dimensional reduction, but we can club together all the neurons in this network that belong to a particular group. And let's look at the mean firing rate of these neurons, right? And what you will see is that at each of these gray lines where we are perturbing the network using some random perturbation, you will see that there are changes in the mean activity of the neurons belonging to a particular node, right? So different communities are going to show different activity, and they seem to go sort of oppose each other. There seems to be some competition between this community. Now, if one thinks about how the order representation evolves in time with each perturbation, what happens here is, so let's say that you have the state of the network in some particular location out here in that's plotted in this two-dimensional space. Now, with each input or each order input, each order pulse that comes in, there can be a jump from that to another state. So you have a whole bunch of different states that the network can visit. The identity or the specific location of these states is dependent upon the connectivity of the olfactory network. And you'll see that the system jumps between these different states as different inputs come in. Right. And what happens, so let's sort of drive this. So this particular network was driven using a periodic input. Right. Now let's drive this using an intermittent input of the sort that you would expect that an insect would encounter if it were to stick its antenna into a turbulent plume of air. Right? So here's the profile of the odor. So there's some probability that the odor is going to be present versus absent, and just flips back and forth. And this is the activity of a bunch of projection neurons that receive this input. Now, when you look at the correlation between the activity of different projection neurons, you find that when you present the system with intermittent inputs, you find these rather clear blocks of high correlation that are formed, and off-diagonal blocks that tell you that the system is has lower correlations, or that these neurons, so 1 to 15, are less correlated with 15 to 30 than they are with. Right, so within group correlations are high in this particular case. Now, we can end up with multiple groups, you can end up with multiple communities, and we've seen that over a lot of simulations that with multiple communities too, the same, more than 2 communities, the same sort of observation still continues to hold. All right, and this is just to compare the correlations between this particular dynamical matrix and make sure that it is does better than a completely shuffled network. Right? So the community structure seems to be a meaningful community structure. All right. So this sort of leads us back to the question of what are the neural invariants of an odor. And the idea that we're trying to get to out here is that different odors stimulate different subnetworks of neurons. And the reason is because each of these receptor neurons are sensitive to particular odorants and less sensitive to others, right? So when you puff an odor on the antenna, it's going to stimulate a subset of neurons in the antennal lobe. And the spatiotemporal dynamics of the network that is stimulated, this dynamics reveals the underlying topology of the network. So the structure of the subnetwork is invariant, right? Regardless of how the odor comes in, whether it comes in an intermittent manner or whether it comes in as a DC input, the structure of the subnetwork is invariant, and that structure is revealed by the dynamics. So it really is the structure of the subnetwork that is an invariant representation of a particular odorant. Now, the question is, why is intermittency a bug and not a feature? Well, one of the things that we noticed is that the input out here on the left is a DC input. So that's this flat input that drives the system. And we changed the degree of intermittency to various levels across a bunch of simulated orders and trials. And what we find is that the intermittency or intermittent input reveals the network structure better than static inputs do. So if indeed the this particular structure is the order invariant, then intermittent inputs have a better chance of figuring out what this structure is. And the second thing is that here on the X and Y axis, we have plotted the mean firing rates of different modules that we detect using the community clustering algorithm. And on the top, we have DC input that's been given to the network, whereas we give input with different levels of intermittency to the other two instantiations of the network. And what you will see is that if you give it a flat uniform input, then the representation doesn't visit a large swath of the space, right? It's sort of restricted to a small region of the space, and intermittent inputs would perhaps reveal then more about the order representation than static inputs are likely to. All right, so everything that I told you so far looks at the activity of the inhibitory interneurons. But then it's not the inhibitory interneurons that are projecting outside the antenna lobe to the mushroom body. It's the projection neurons. So whatever we see in the activity of the inhibitory interneurons must somehow be reflected in the activity of the projection neurons for it to have some effect on further processing, on further olfactory processing. And the question is, how do we figure out whether the projection neurons indeed show some of this community structure? And the way we sort of thought about this is, let's look at this particular example where, let's say that we've detected 3 communities, and each of these communities would be synchronously activated or would have highly correlated activity when presented with a particular odor. Now, if we look at the projection neurons, we can find out how many inputs it receives from each of these communities. So this particular projection neuron receives 2 inputs from community 1, 1 input from community 2, and 0 inputs from community 3, right? And in a space that is determined by the inputs that the projection neurons receive from different modules, you can locate this particular projection neuron in the space at some particular point in space. Similarly, another neuron could be located at a different point in space. Now, when you look at the activity of projection neurons, the input that they receive from the inhibitory interneurons seems to So projection neurons that receive very similar input from inhibitory interneurons is going to do qualitatively the same things, right? So in a sense, the mapping, the distance between projection neurons out here should also be related to the kind of activity that they would generate. So here is the distance matrix between projection neurons in this particular space. And you'll see these 2 blobs, which says that the distances are high, and along the diagonals, the distances are 0, and so on. And when you look at the activity of the projection neurons, we look at the distance, not the correlation in this case. We looked at something known as the von Rossum distance, which is the distance between spike trains of individual projection neurons. But what we find is that the matrix that tells us about the structure or the location in this particular space strongly correlates with the dynamics, with the distance between the spike patterns that the projection neurons generate. So the idea out here being that whatever modular structure that you see, the modular structure that you see in the inhibitory interneurons, you can see a reflection, or you can detect that modular structure in the activity of the projection neurons as well. All right. So I'll just a couple of takeaway points before we end here. One is that it seems that active sampling is a way to explore the state space of the network. And initially, we thought that this might, in fact, hasten the ability of the system to differentiate between odors. But we are more, based on our simulations, we think that it seems like it fills in the details. It helps you discriminate between very similar odorants by using this intermittent sampling. And the second is that, in a sense, the network structure is itself the odor code, right? So the network structure, the structure of the network that gets stimulated when a particular odorant is presented, is in fact a good proxy for the odor representation itself. All right, so I'll stop here, and thank you all for listening. Questions?
Thank you very much, Collins. And you have been remarkably well in time. Collins, is your usage of the word intermittency, or how similar is the use of the word intermittency in your context to what we normally consider intermittency in dynamical systems?
You know, I should say that I've used it somewhat loosely over here. So here it's just random on-off patterns of activity.
No, I mean,
right?
there's a very well-defined sense in which most dynamics people would use it, which is, you know, that there are these events, and one can see the events separated by certain time intervals.
Right.
And if the time intervals have a certain pattern,
right,
then one tends to call it an intermittent.
Right. So here the it is just randomly chosen. So I should have perhaps used the word random input rather than intermittency, except that the random input is on-off kind of patterns.
No, but maybe there is something more to it. Speaking of which, what are the time, you know, you've presumably done Fourier transforms of these signals, and have you examined the power spectrum?
So these are inputs that we are constructing. These are not inputs that we are measuring from actual plumes of odorants. Right. So the way we are constructing this is essentially just using a random number generator and deciding whether it's going to be on or off. Right. So here I don't expect to see anything dramatic with the power spectrum. However, in an odor plume, there are certainly some frequencies that are better represented than they would be in the signal out here. So we haven't looked at multiple time scale phenomena. I think
it could even be valuable to take your input stream and look at its power spectrum and look at the response, and you know, there's some correlation between your input and your output.
Yes, certainly there is a correlation between the input and the output, because it's when the input comes in that the network is activated. And if you look at the broad, so if I were to take the sort of firing rate of all the neurons put together, I would actually be able to recreate the input that's being presented. But to me, what is interesting out here is that even though the average firing rate would give me would just completely reproduce the input, most likely completely reproduce the input, it's how the state switching happens that is really interesting. And which neurons fire synchronously in each of these pulses that come in that reveal the structure of the network. So on average, it would reproduce the input, but the details will tell us more subtle aspects about the network.
If you'll ever do that, I'd be interested in learning about how the correlations come about in that.
Yes.
Okay. There are questions in the chat.
I see Vidita has her hand up.
Yeah. Is it okay to ask, or maybe you can wait till you finish the questions?
Well, I was just going to read out, Rituparna's question to everyone. Are the single projection neurons tuned for a particular order, or is it only a subset of networks that are tuned to a particular order? Is there a presence of one-to-one mapping in this case?
Oh, so is a single projection neuron tuned? No, so typically so here's the way it goes, right? So a receptor neuron will have different sensitivities to a broad array of odorants, right? And a projection neuron that gets input from that particular receptor neuron, if there is a glomerular architecture, will show similar sensitivities, barring lateral inhibition that you can also see. So a single projection neuron can respond to multiple odorants. It's not a one-to-one mapping between odorants and projection neurons. Does that address your question? Sorry.
Rituparna? That was actually a question in the chat box, Collins.
Okay. All right.
Okay. Any other questions?
I had a question, if I may.
Yes.
Yes. Hi, Vidita.
Hi, Collins. Nice talk. I was wondering, in a system in which you have turnover of the odorant receptor neurons, like you have in vertebrate odor systems.
Yes.
What does this do to the stability of the network at the level of the glomerulus, given that you have this much, 90-day turnover? So what is the thought on that?
So it's actually quite, that's really quite amazing that it remains stable despite this kind of turnover. So of course, the neurons that continue to express the same odorant receptor will continue to go and innervate the same set of mitral cells in the case of projection neurons, and in the case of mammals, and projection neurons in the case of insects. So despite the turnover, they're all going to hit the same place due to, of course, various guidance mechanisms that are present, which guide these axons to the right place. So the representation is certainly stable despite the turnover.
So in a sense, would you think about it as having stable engrams in terms of long-term for holding odor-related memories as well at the level of the olfactory bulb as well? Is it that stable? It seems too hardwired to leave flexibility for variations. I was wondering what your thoughts on that.
Okay, so so there is a certain amount of hardwiring, certainly, right? From the receptor neurons to the glomerulus, that seems to be quite hardwired. Vidita, I don't know how much of the answer you...
That's okay. We can chat about it later.
Okay, all right.
Yeah.
Okay, Collins, there was a question from Sarthak and one from Rituparna.
Okay.
So, Rituparna, would you like to go?
Yeah, hi. So the one that I had asked in chat box was with reference to the fact that we have these place cells in hippocampus, which are like you have a spatial tuning to brought down to a single neuron level. So I was asking in that context that you have these excitatory neurons in the olfactory lobe that are very particularly tuned for for a single odor, apart from having network.
Right. So the projection neurons are also not tuned for a single odor. They can respond to multiple odorants, and they can also respond in different ways to multiple odorants. So it's not just whether they don't respond to just a single odorant.
Okay, yeah, thank you.
Sarthak, please.
Hi. So I guess, so my question was, I mean, can I think of this in some information theoretic way? Like, is there some sort of information advantage to be able to sampling things intermittently as opposed to just trying to directly sample the entire DC stream? Because I mean, I can see like in the particular network model, there is some advantage in terms of unveiling the underlying network structure. But information theoretically, is there some way to think about this?
So in the network model specifically, the reason that you sample intermittently is because it lets you visit different states of the network. And that in turn helps you build the representation over time as you're sampling multiple states. Now, there are, you can sample intermittently and perhaps navigate to an odor source. So there, certainly intermittent sampling would help. In fact, there is a study that looks at how you, I guess one could say, gain information by sampling, because it doesn't have to sample intermittently. The input itself is an intermittent input. So how can one use that to gain information about location, for example, that you can navigate to? So certainly intermittency does seem to play, not intermittency, I should say, input that comes in a pulsed manner certainly seems to play a role in that.
Yeah, but I mean, I guess, I mean, what you were showing is that even if you presented a DC plume, the insect seemed to prefer an intermittent sampling. So it seemed like there might be some computational advantage to doing that.
Right. So here So here the computational advantage is that having a DC input makes the network get stuck in a particular state. It doesn't jump out of that state. And having intermittent sampling helps it jump out of that state and go to a different state. Does that seem reasonable?
Yeah, I think it does. And I think maybe we can discuss this more later also. I don't want to just hold everyone up. But I guess then I was also thinking that, does it then seem that if I'm looking at any kind of memory model where I'm trying to represent, say, various inputs via, say, clusters of activity in some sort of neural substrate, would it seem like it might be advantageous in general to be able to get that input intermittently rather than just constantly receiving the input?
So, I think about this clusters of activity, right? Whether, so this is, I should separate the question into two parts. One is the part whether intermittency itself has intermittency has advantages. And the second is whether the synchronized activity is, whether that plays any specific role. So in the insect olfactory system, if you look at neurons that are there in the mushroom body, the Kinnean cells in the mushroom body, they're exquisitely sensitive to the synchrony in input. So one could think of the mushroom body as being some sort of a detector of modules that fire in a synchronized manner. But as far as now detecting, so if you don't have intermittency, if you don't have these pulses of input coming up, then once a particular module is activated, it's going to remain active, right? It's not going to switch to another module. So you don't get the entire structure of the network unless you have pulsed input coming in. And there's a detector further down that is specifically designed to detect this kind of a synchrony.
Okay, yeah, thanks.
One second, there seems to be something in the chat. All right, does that bring us to the end of the questions? Collins, we're going to see you in a breakout session tomorrow, is that right?
Yes, I
Yeah,
think that's
you and Vidita and so on. So we'll have a chance to catch up. And this later today, when we chat with Sarthak, I'll ask him about this network inference techniques that he's been applying and so on. And it will be really cool to find out whether your intermittency as colloquially understood has something to do with the non-colloquial understanding of intermittency as well. Okay. Thanks a lot. Thanks very much, Collins.
Thank
And we'll be back in a short while for Vidita's talk, almost about an hour or so. Okay, so look forward to the next talk.